Okay. So they need a very solid cylinder at the center so that it will resist the compressibility of the roots. Then we are also saying that in the leaves, they are also located at the upper side of the midrib, and that enables the leaf to be pushed, but it resists the tearing or tearing forces that may act on the leaves and then the blade. And that's why the leaves remain compact, even if there is a very huge stress that is brought about by the flowing wind. And then also in woody stems, they are actually formed from mainly the lignified xylem cells. And these lignified ones tend to make the stem very hard and very rigid. And therefore, they will not now need to rely on the turgidity of the cells, but those tough tissues are able to withstand any tensile stress that may be caused. So what about aquatic plants? Because we've talked about support in terrestrial plants. The aquatic plants, sometimes we call them the hydrophytes. So the hydrophytes also have a challenge that they can be blown by the wave of the water. They can also be washed away by such plants. They are not anchored or rooted in the soil because all the time they are washed. So how do aquatic plants get their support? By the way, aquatic plants don't need a lot of support because for them, the water that they are in is much higher in terms of the density than the air. So it provides a great upthrust force and therefore maintains them in their position. And then you will find that in aquatic plants, they have a lot of intercellular spaces, large air spaces in between them. And that intercellular space is usually air filled. And therefore, it goes into their tissue and ensure that there is buoyancy. Buoyancy means that they're able to float, they're able to move with the water current without it getting distorted or broken down in any way. So in terrestrial plants, unfortunately, when you remove them from that water, remember in water, being more dense provides the buoyancy, and the plants also have some large intercellular air spaces which are filled with air and therefore makes them lighter so that they are able to float and then come at the surface of the water so that they can maximize uptake of sunlight, and then they are able to photosynthesize. But the worst thing is that the moment you remove this plant from water, they will be able to collapse very fast because they lack those mechanical tissues we were talking about. They lack the collenchyma, the sclerenchyma. Instead, they are replaced by intercellular air spaces that ensures the buoyancy, that ensures that such plants are able to float. For example, water cabbages, you find that they have hollow rods in their stems that is air-filled and makes them much lighter so that they can float on top of the water. So you'll find that even their xylem tissues are very much reduced. First of all, they don't need a lot of water because they're already in water. Then secondly, because they have very thin cuticle, they can actually absorb their water by the process of osmosis. And then three, they don't need to support themselves so much in terms of the rigid pressure of air and probably animals that come. So in that regard, they have poorly developed xylem tissues. And therefore, if you remove them and put them, because of that lack of those vascular tissues, they lose their water quickly, and then eventually they collapse, and probably they will be able to die. So when you look at this, we look at the comparison in terms of support in terrestrial plants and hydrophytes or aquatic plants. One is that the terrestrial plants require mechanical support because air will not hold up the plant structures in the way water does, because air is less dense and therefore does not provide strong, buoyant, and upthrust that can keep a plant in position. But in aquatic plants, the density of water is much higher than air, hence providing a larger upthrust to the plant. And therefore, the plant will be able to support itself or even float on top and be able to gain the necessary light that it will require. Number two, in terrestrial plants, there's presence of collenchyma and sclerenchyma cells, which has an abundant lignified thickened walls. And this means that their support depends entirely on these specialized thick-walled cells. But here, no collenchyma cells in a hydrophytes, no sclerenchyma, poorly developed xylem. So all these indicate that aquatic plants do not depend on the cells for mechanical support, but rather they rely on their structures being air-filled and the buoyancy of the plant. Then you'll find that in terrestrial plants, there is very small air spaces in the stem since the stems have low density. So they only provide limited support. The air spaces in the stems and then in the leaves provide very little support. But in aquatic plants or in hydrophytes, they have large, numerous air spaces in their stems and the leaf. So this suggests that their support will depend mainly on the buoyancy, the ability of it to float, the ability for it to float on top of the more dense water, such that they can offer their support, they can maintain their position and absorb as much light as possible. So members, let's look at a sample question that was in UNEB that tries to use the information that we have. You can copy that question. The first one was saying outlines the importance of support in terrestrial plants, which actually we've talked about. Explain the mechanism of support in dicotyledonous plants. I think we also talked about that, relying mainly on the tagger pressure, relying on mechanical tissues, and then relying on some of those tissues. The question is, what if the terrestrial plants are put in water? Will they be able to survive? The answer is no, or for a very short time. Why? Because this terrestrial plant with very limited air spaces, because they are less buoyant. And because of that, they will sink in the deeper part of the water. They will not be able to absorb sunlight. They may not be able to take the necessary air or oxygen that they need and carbon dioxide for photosynthesis. So because they are not buoyant, they cannot float in water. Therefore, they sink, and then their tissues. Yes?
What I meant is that if the aquatic plants are placed in a waterlogged area like a swamp, and they also still surviving.
Areas lack that aerial air or oxygen. And that means that they do not really have that air which is blocked or locked within their tissues. And because of that, they will not be able to respire. The roots will not be able to respire. And secondly, they will be able to absorb too much water that may actually cause dissolution of the food that they have made, and therefore they will not be able to have enough nutrients. And then in waterlogged area, there's also a lot of leaching. And this leaching ensures but in plants that will be able to survive better, because for them, they are able to obtain that nutrients even in the water because they float in the water. They don't sink down to the basement of the water where the minerals are leached. So terrestrial plants don't really survive well in waterlogged area. They survive mainly in dry, semi-arid areas with limited water and only moisture, and not the waterlogged areas. Okay, any other question?
So
get this question.
The question of
Davis, your network. Davis, can you hear yourself?
Referring to the question of Prosper, where I was asking whether the aquatic, Jesus, to the question of Prosper, where he's saying, meaning, okay, like aquatic plants that majorly reside on lakes, beaches. Yes?
I had not seen the question. They are saying, what if the aquatic plants are placed in waterlogged areas like swamps? Can they survive? Yes.
Yeah.
Waterlogged areas, waterlogged areas is the same as organisms that are staying in aquatic habitat. Yes, it's true. I thought you were saying if terrestrial plants were taken to a waterlogged area. Yes, Pesh, you had your hands up.
Teacher, I wanted to ask about the plants that wilt even when they are shifted from an aquatic like area to an area that is less aquatic.
That wilt?
Yes, like those plants that wilt, they're aquatic plants, but they wilt, for example, in swamps. I wanted to ask about that.
Actually, me, I've told you that there are many other factors that are in play, especially for plants that wilt. Sometimes they are wilting because, especially those that are rooted plants, usually wilt because once they are rooted, then their roots will not be able to obtain some of the nutrients they need because they are leached. That is number one. Number two, there is less air in the water, meaning that the roots will not be able to respire. But for real aquatic plants that are buoyant, sometimes their roots come off the water. You talk about that kind of root, the aerial roots come, such that they are able to respire with the atmospheric air, not in water per se. Somebody was putting the hand. Is it Davis?
No.
Usually wilting is a result of loss of water, but there is that element where tissues die, either of taking excessive water or either because the cells cannot be able to obtain adequate nutrients because the plants are not getting enough water to photosynthesize, or they are deficient in certain minerals. That is what happens when they are in aquatic habitat. But if it is related to loss of water, which is tissue desiccation, it means that it is only plants that are taken from well-watered place, waterlogged area, or in water that is taken on dry land. They are the ones that are going to lose their water very rapidly because they don't have those tissues, mechanical tissues developed to withstand that high intensity of desiccation. And because of that, they start wilting. But the ones that you're seeing drying in water is not related to the loss of water. It is inadequacy of the nutrients, inability for them to photosynthesize, and lack of air or oxygen required for respiration. So some of the tissues die. And once they die, you begin seeing the plant drying off. And then eventually it will collapse and die. Okay, have you seen those questions? Outline the importance of support in terrestrial plant was carrying 4 marks. And I think we talked about that. The importance of support in terrestrial plant. We talked about enabling them to hold their leaves, exposes the flowers, holding the fruits and seed for dispersal, and then maintaining the shape. Then another question was that you explain the mechanism of support in dicotyledonous plant. I have told you dicotyledonous plant rely mainly on those supportive tissues, the mechanical tissues that are usually distributed. Like now in young plants, they are at the periphery, they are at the edge, and then in the roots, they are found deep at the center of that. In the leaves of dicots, they are mainly found at the mid-ribs of these leaves. Those are mechanisms for support. But the main idea behind all those that are herbaceous dicots is that for them, they rely entirely on the turgidity of their cells, such that they stretch against one another and then causes the stiffness of the stem, and therefore it will be able to stand erect. Then they're saying, describe the different mechanical tissues found in plants. How many mechanical tissues do we have? We have mainly the collenchyma, which they want you to describe. You have studied them in your histology. And then we have the sclerenchyma. And we have also discussed here how they are distributed and located. The collenchyma have uneven thickened cellulose wall. The tissues provide flexible support to the stems and leaves. The walls of the pressure and tension. That is what they want you to describe. Then you come to the collenchyma. Also, you describe their location, their structures, and then what they provide. That is what that question requires you to take into consideration. Then another question is saying, describe how the distribution of vascular tissues is adaptive to its function. We also talked about this, the distribution for the young dicots, distribution in the roots, distribution in the leaves, and then distribution on mature stems in terms of the secondary xylem, which actually makes the woody plants very stiff. That is also what you are expected to discuss. We talked elaborately about the distribution. We talked about distribution of vascular tissues in young plants. We talked about them in dicot plants. We talked about them in leaves. We talked about distribution in woody stems. That should also be able to answer that particular question. Then they are saying outline the support in terrestrial plants, and that outline, support in terrestrial plants and that in aquatic plants. I think here we are talking about the same things that we talked about here. You compare the support in terrestrial plants with those of the aquatic plants. That differences would be outlined, and then it shows how terrestrial plants support themselves and how aquatic plants should be able to support themselves. So you'll be able to, should be able to read and try to relate them to the questions that are always examined. Now we are going to look at locomotion and movement. Like I'm saying, locomotion means changing position by the entire body. All animals, actually all animals move, and they displace the entire body from one position to another. And we call them organisms that locomote. Then movement is the act of displacing parts of the body while maintaining others. Now, the study of movement is called kinesiology. It is from the word kinetic, moving energy, moving form. So it is called kinesiology, but we are saying for locomotion, there must be entire body moving. Then movement is the act of just displacing any part of your body. Like for me, my hands can move, but you can't say your hands are locomoting because your hands have a fixed position. They just change positions, they just displace themselves. So your hands can move, your eyes can move back and forth. Okay, somebody is saying it doesn't have the PDF. I think I will send after the lesson. For those of you who don't have, me, I'm assuming all of you have the notes that we are talking about. I'm just trying to pick important points and discuss. But I think I sent this information long time ago. So you should be having it. Those who don't have it, I will share on the WhatsApp, and then you should be able to have it. So I'm just picking the key points of that, but there are many other information that you have there, which I can't bring in the slides for us to study all that. Now, we are going to look at movements. Then we talk about locomotion after that. Basic types or mechanisms of movement. One is called the ciliary movement. The second one is called the Amarboid or flagellar movement. And then the third one is called muscular movement. These are the three basic mechanisms of movement: the ciliary movement, the amoeboid movement, or sometimes we call the flagellar movement, and then we have the muscular movement. So we are going to start with amoeboid movement. Now, in amoeboid movement, you are talking about that kind of movement characterized by temporary protoplasmic protrusion to form a temporary feet-like structure called a pseudopodia. Can the word outline be answered similarly as to compare when answering question 2B? What is question 2B? 2B is explain the mechanism of support in dichotomy. 2B is outline the support in terrestrial plants. And so outline means that you are not going to the details of it. So you can actually put them in a table, you compare them, or you can give it as an essay where you talk about movement in terrestrial plants separately. You also talk about support in aquatic plants also separately, but you can also give them in table in terms of the comparison, because you are just outlining. You are giving some basic fact or basic information about that. So let's look at amoeboid movement. Just like I've told you, you have amoeba. I'm trying to get something to draw that. So you have an amoeba. Remember, amoeba is shapeless. Of course, with its nucleus. What happens is that this amoeba starts developing a feet-like structure called a pseudopodia. As it is developing that, you find that this part starts moving and then the leg remains there. Then you'll find again another part will develop, leaving the leg-like structure. So as you continue with that movement, you find that it is changing the position from here. With the development of this pseudopodia, the whole body moves to the next stage. Another body moves, so that by developing this leg-like structure, which are actually not legs, but they are leg-like structures, they are able to displace themselves from one position to another. That is what we call the amoeboid movement. So amoeboid movement means a crawling-like type of movement, which is characterized by protoplasmic. Part of the body expands. Part of the body expands, what we are calling a protrusion. And then it develops a temporary feet-like structure, and that structure is called a pseudopodia. We are going to look at the relevance. Why is amoeboid movement important? Two main areas. Number one, it enables amoeba to move about to obtain food, to avoid dangers, and to escape from the enemy. You might think the speed is too slow, but of course that slow speed enables it to escape from certain organisms. Number two, another organism or a cell that exhibits amoeboid movement is the white blood cells called the leukocytes. So we have the phagocytes, we have the macrophages of the limbs, and the Kupffer cells of the liver. All of them undergo what we call amoeboid movement. But for them, all of them, they engulf an antigen or a microbes, and because of that, they are capable of destroying those microbes, and then they immigrate in the circulatory fluids. They move from wherever they are into the circulatory fluids like the blood and then the limbs, such that they're able to move using that phagocytic method, movement, using that kind of protrusion that enables them to move. So those are the main relevance of amoeboid movement. For amoeba, it can move to obtain food, to avoid dangers and escape from certain energy. While for white blood cells, like the phagocytes, the macrophages, the lymphs, the Kupffer cells, for them, they use amoeboid movement to engulf the microbes and also to immigrate from the circulatory fluids. Immigration means that moving out of those circulatory fluids, and then they go into the cells and then enter or penetrate the tissues that they eventually infect. So we are expected to describe the mechanism of amoeboid movement. Of course, we have several theories that talks about amoeboid movement, which I'm going to share with you briefly. One of the most popular theory is called the Sol-Gel theory. The Sol-Gel theory, and okay, there's some. So I'm sharing with you briefly so that you can experience firsthand from how amoeba moves. Sorry. So look at that.
The cytoskeleton is a dynamic network of filamentous proteins that extends throughout the cytoplasm, forming the struts, cables, and girders that give.
So you can see it's developing new structures. Let me put another one. Look at this structure, how it is changing. Now you can see that there is a fluid part and then a solid part. This is the solution side and this is the gelation side. So you're seeing that there are a lot of molecules that are interchanging within this or entire component. You can see the solid now turning into a liquid and then pushing the pseudopodia. Then this one again is flowing back. So this is the solation side. This is the gelation side. Now the fluid is going to flow in this direction. By the way, this one is moving in this direction. So the fluid flow in front, and then at the same time, this one moves backwards. And then a new pseudopodia is formed. Okay? If the pseudopodia is formed, then the whole body is displaced. Look at this one. The solid will move here, result into formation of a new pseudopodia. So that is the appearance that talks about a last one, which is we will use it to explain that. So
rate of metastasis. While several hypotheses have been proposed to explain the mechanism of Amarboid movement, the exact mechanism is still unknown. Based on some mathematical models, recent studies hypothesize a novel biological model for collective biomechanical and molecular mechanism of cell.
Members, are you able to see?
It is proposed that microdomains weave the texture of cytoskeleton, and their interactions mark the location for formation of microdomain signaling dynamics organizes microdomains trigger and maintain active polymerization of that continuous application of stress. Propose that the cell cortex ectomizes and contracts to increase height.
Okay, so let's get back. And then we discuss it is getting stuck somehow. I don't know why I can't share. Joseph, first help us with the sharing. It is getting stuck. I don't know why. Members, are you seeing? Are you reading my mechanism?
Yes.
Yes.
The one on my chart. Eh?
Yes, we can see it.
I'm struggling
Yes,
to share.
Yes.
So you have seen where we are talking
Yes.
about mechanism of Amarboid movement. Now, just like you have observed, we are saying there are many theories about Amarboid movement, but then the most common that is accepted in our Ugandan context and among most of the biologists is what we call the Sol and Gale Sol theory, or transformation of the cytoplasm. And this was invented by a man called Mast in 1925. So we are going to discuss what the theory postulates. Now, that theory has not yet been confirmed. It is not the only, there is a lot of research that is going on. But when you look at this, you'll find that when you look at amoeba, there is what we call the plasmalemma. This is part of that direction of movement. Now, there is an hollow part at the sides that you were seeing moving, which we call the plasma gale. And then you have the middle part of this. And it is this middle part of this that keeps on moving, and then it pushes the upper part. Now, the gale means that it is semi-solid. The sol means that it is a solution form. So you are going to find what is going to happen here is that this solid starts to melt. Once it starts to melt, it flows forward. Now, as it is flowing forward, it creates a tension here. Now, as it flows, it stretches the cytoplasm, and therefore you'll find that the plasma gale or the plasmolemma that bases itself on the substratum will expand. Now, as it moves forward, then you'll find that this solution starts flowing back because there is an inner layer here that allows the solution to flow only from inside. While you see that as it comes out of this into the plasma gale, then this solution starts moving backward. Now, as it is moving backward, it pushes this forward, and yet this one here, the gale here, continues to enter. So as it moves here, as the gale enters and this one is moving forward, the whole organism keeps on moving, with this one flowing, forming a new